The phenomena that we will discuss in this last section of our lecture is cytokine storm and the life-threatening complication that it can lead to. Cytokine storm, or hypercytokinemia, is a severe immune reaction in which the cytokine production is not linear, but many cytokines are produced by the immune cell at an accelerated rate that disturbs the balance of the immune system. Although cytokines are crucial for healthy immune responses, an abrupt increase in their production might be dangerous. An infection, an autoimmune disorder, or another illness can induce a cytokine storm. It may also occur after treatment with some types of immunotherapies. Signs and symptoms include high fever, inflammation, severe fatigue, and nausea. Multiple organ failure can occasionally result from severe or life-threatening cytokine storms, referred to as hypercytokinemia. If we look at the mechanisms of how the cytokine storm occurs, we must recall some basics of the immune response. Now, as mentioned before, cytokines are produced as a result of immune cell activation, and in turn, they stimulate more immune cells. An example of this positive cycle would be that of Th1 cells. They produce interferon gamma and interleukin-2, which stimulates more Th1 cells to produce more interferon gamma and interleukin-2, and the cycle continues until it's needed or T regulatory cells break the cycle. In case of severe infection or autoimmune disorders, what happens is that the cycle continues without any regulation, and more cytokines are produced at a higher rate. If we take the example of dengue fever, when the pathogen causing dengue fever enters the body, it stimulates immune cells such as the macrophages and dendritic cells. Once the virion enters, it is picked up by receptors on the monocyte, where the virus replicates and more copies of the virus are made. Once the population of virus cells increases, production of type 1 interferon begins. The type 1 interferon, which is interferon alpha and beta, signals the nearby cells to increase the expression of MHC1, decrease the production of protein, and endocytosis, so the virus cannot replicate at a higher speed. The second pathway or step that takes place after the virion penetrates the first line of defense is that it gets recognized by the immature dendritic cells. The immature dendritic cell binds to the virion and moves to lymph nodes, where it changes into the mature dendritic cell. Then it presents the viral antigen to CD8 and CD4 T cells. In response, CD4 T cells produce IL-2, which stimulates the CD8 T cells. They proliferate and grow and leave the lymph nodes to fight off virally infected cells. As the virally infected cells start to die, nearby monocytes and macrophages phagocytose these cells and become activated. They may also become infected if they pick up a live virion. So, to put it simply, the immune response is initiated in two ways. Live virions infected the immune cells, leading to the production of cytokines, and dead virions get phagocytosed and leading to the activation of macrophages. We can also conclude as being one arm of cytokine activation through the innate immune response, where cells like macrophages, natural killer cells, and dendritic cells are activated and produce cytokines like interleukin-2, interleukin-6, interleukin-12, interleukin-beta, and TNF. All these cytokines are very potent. They make the endothelium of blood vessels more permeable. Other cytokines, such as type 1 interferons, are also produced by the Th1 cells along with IFN-g. They further activate the macrophages. More and more cells get activated, more and more cytokines are produced. Remember the cycle we mentioned before. So, the magnitude of the immune cell response increases with the production of more cytokines. Eventually, the balance is tipped and the body goes into crisis. Since many cytokines are pro-inflammatory substances, excess in their production can lead to excessive inflammation. One example of the cytokine storm leading to life-threatening conditions is found in patients afflicted with COVID-19. Recent research on patients with COVID-19 infection has revealed that these people have significant amounts of chemokines as well as pro-inflammatory cytokines such as interferon gamma, interleukin-1 beta, interleukin-6, and interleukin-2. The cytokine storm has been utilized, like many other viruses, as a warning indicator for clinicians to identify illness acceleration, especially for SARS, MRSA, and influenza. In the absence of treatment, the cytokine storm caused by COVID-19 results in immunopathogenic damage that, in many cases, not only causes ARDS, but also can advance to severe tissue damage, organ failure, and death. If we look at the ARDS, or acute respiratory distress syndrome, a bit more closely, aside from these viruses, ARDS can also occur because of pneumonia, sepsis, pancreatitis, and blood transfusions. The pathogenesis of ARDS begins with inflammatory damage to the alveolar capillary membrane. The cytokines that we discussed in the previous section play their role to cause this inflammation. Like any other form of acute inflammation, the permeability of the surrounding vasculature, which in this case is that of the lungs, occurs. As the lung permeability rises, protein-rich pulmonary edema fluid is drawn into the lungs and plaques form in the blood vessels, ultimately leading to respiratory insufficiency. Normal blood flow, as well as the oxygenation process, has been halted. So, you have seen how the excess production of these small but potent cytokines can lead to multi-organ distress. So that is all about cell communication and the cytokines that we will discuss in this lecture. If you wish to learn more, check out our video on the cytokine network.
And keep watching sqadia.com for videos with plenty of knowledge.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.